Hello, everyone, and welcome to the webinar entitled Manual Control of Phragmites, a Pilot Study, hosted by the Federation of Vermont Lakes and Ponds, also known as FOVLAP. I'm Jerry Bergdahl, FOVLAP board member, and FOVLAP since 1972 has remained dedicated to fostering water quality standards and to help preserve Vermont lakes, ponds, watersheds, and aquatic ecosystems. Please consider joining us with individual memberships starting only at $25. We'll add a link in the chat. Next week is National Invasive Species Awareness Week. You could check out the website, and Phragmites is definitely an invasive of concern. Today's presenter is Annie Dalton, avid amateur botanist, citizen scientist, and volunteer with the Lake Champlain Committee Cyanobacteria and also aquatic invasive species monitoring programs. Annie will leave time at the end for questions. You can use the raise hand feature or put your question or comment in the chat. Please stay muted until you'd like to speak. We'd love to know where you are zooming in from. Please write down the town you're calling from and the lake you represent or frequent most in Vermont in the chat. And now I'll hand it over to Annie, but there is the problem with everyone being Pat right now. So you can rename your to tell us who you really are by clicking on the upper right-hand corner and using the drop-down menu to rename. Now, thank you, Annie, and we're anxious to hear your information.
Great, thank you, and welcome everyone. Good afternoon. I want to give you a little information about my background and my interests. I've been fascinated by plants all my life. I received a bachelor's and a master's of science degree in botany from UVM, where I had the good fortune to work under Bud Etherton, who taught me plant cell electrophysiology and encouraged me to act on my curiosity. After a couple of lab tech jobs and volunteer teaching, I found that I was better suited to organizing information, customer service, and teaching computer skills. So I worked at the Fletcher Free Library for the past 20 years and retired in 2023, keeping studying plants as a hobby. I take photos of plants, mostly close-ups. I'm fascinated with the structures, and I use them to make bookmarks, greeting cards, and slideshows, which I share with people, hoping to inspire that sense of wonder that I have about plants. And my recent botanical focus has been on aquatic plants. Last summer, I decided to apply my skills to benefit the community, both by addressing Phragmites in Nichols Pond and also as a volunteer for the Lake Champlain Committee.
Excuse me, Annie, I just wanted to mention that someone found your volume low, so maybe just speak up a little bit more.
If you could. Okay, will do. So this presentation is designed to be standalone. So you're going to find some of the slides will have a fair amount of text on them, but please don't feel it's necessary to read that text because I'm going to be replicating the text in my narrative. And because I have a lot of links to other slides, these are links to slides on the show, and these are links to slides that have resources with more links on them. So that's why those will be available in the PDF file that's going to be saved on the FOVLAP website after this presentation. Let's see here. Okay. So to introduce the project, Phragmites australis subspecies australis is a non-native invasive grass, probably originating in Europe and brought to North America in the 1800s. It has been spreading in Nichols Pond in Woodbury, Vermont since at least the mid-1990s. I fell in love with Nichols Pond 30 years ago and have been renting cabins at Cahagan ever since. Before it destroys more habitat for native plants, fish, and other organisms, I decided to try a couple of hand removal control measures that I read about in Northern Woodlands magazine in 2024. So first, I confirmed that the Phragmites taking over the shoreline in Nichols Pond was indeed the invasive non-native subspecies and not the native Phragmites australis subspecies americanus. Next, I obtained permission from the landowners of Cahagan, the Meyer family, to harvest along their property's shoreline. Then I checked with the state of Vermont Department of Environmental Conservation to make sure that manual harvesting would not require a permit, and it does not. Finally, I chose a few sites along the shore of Cahagan property to conduct a pilot study, small enough sites so that I felt I could commit the time and energy to harvest the recommended 2 or 3 times during the summer. So here is an example of the natural shoreline ecosystem at Nichols Pond. I took this photo in 2012. I used to go up there every October, and I found later that I had actually taken a photo from approximately the same spot many times. So I was able to look back and see when Phragmites started infiltrating this area. So at this time, the shoreline vegetation and in the littoral zone is mostly, appears to be native plants. However, if you look right down here in the lower right corner, there are a few Phragmites starting to grow right here. Phragmites causing loss of shallow water habitat. There's great blue herons on Nichols Pond, and they hunt in the shallows for crayfish and other things. The fish tend to nest in the shallows, and loons on the pond enjoy eating the young fish. And turtles, there are turtles in the pond, and they require access to the shoreline to be able to get out and nest and lay eggs. There have been instances where turtles, not in Nichols Pond that I know of, but have been trapped in dense Phragmites stands. There are also negative impacts for human enjoyment of lakes and shorelines. Here's an example of view loss. So here's some Phragmites growing at Nichols Pond, and in July, and then after harvesting, I've removed them and you have a better view. It's a good time to note that the genus name Phragmites is from the Greek phragma, or fence, testifying to its ability to form an impenetrable monoculture. Another negative impact for human enjoyment is potential blocking of lake access. So here is a dock that's used by the Outspan cabin at Cahagan. And you can see over here on the left, Phragmites has been marching toward the dock from left to right. And if this population hadn't been harvested, it's possible that eventually it would block access to the lake from this dock. So Phragmites has several means by which it can succeed in outcompeting other plants, such as being tall and fast growing, shading shorter plants. It has rapid and dense rhizome growth. The rhizomes are underground stems that grow horizontally and produce vertical stems. And it also produces allelopathic compounds, which suppress growth of other plants. It's a real nutrient hog, sucking up all the available nutrients, and it sometimes can make the soil more acidic, which can be detrimental to growth of other plants. So I wanted to be open-minded when I approached this project. I wondered if there are any benefits to Phragmites. And here you'll see a muskrat in Nichols Pond that had been nibbling on the young Phragmites shoots. We had hoped he would destroy the population, but he really didn't have any significant effect on it. The plants, in fact, have a low nutrient value, so it isn't eaten much. So it does not benefit the ecosystem by being a food source. What about some other kind of nutrient source? I found an article showing that decaying stems in the water release nutrients that stimulate production of or growth of bullfrog tadpoles. Native to eastern North America, the North American bullfrog is common in Vermont, but not out west. It tends to be actually invasive out west. So this benefit could present a double negative ecological impact in Western North America, where Phragmites is also invasive, as well as the North American bullfrog. How about habitat? I wondered. So some birds use Phragmites for cover and nesting, notably the red-winged blackbird and common yellowthroat. However, once the stands become mature and really dense, bird use diminishes. And many of the insects that use Phragmites for larval development are in fact non-native. So its value as habitat for native species is low compared to that of native plants. This shocking photo is on the shore of Lake Huron, and there are really dense populations of Phragmites around the Great Lakes. I thought I'd better show that to show what can happen. So the other potential benefits of Phragmites I looked into are as an artist material. Yes, it has been used for many years. And in China, they make paper from the pulp. And in some situations, it's considered beneficial by stabilizing soils to prevent erosion and also sequestering nutrients, heavy metals, and carbon. This could increase water quality, but colonies need to be harvested and controlled to realize these benefits without experiencing the negative effects of uncontrolled spread. So to sum up, the loss of biodiversity is generally believed to outweigh any benefits of uncontrolled Phragmites populations. So here's the article that provided the initial inspiration for me in Northern Woodlands magazine, and it mentioned two methods of manual control. One is spading, and the other is for underwater plants, cutting to drown the plants. And they mentioned Canada's Invasive Phragmites Control Center. So I started there online to do some research. And I found that there were several organizations that provided a wealth of information on their websites concerning the plant's life cycle, the problems it causes for natural ecosystems and human enjoyment of them, and the control measures that have resulted in some success. Manual control methods are the least expensive and disruptive, but labor intensive, so suitable for smaller populations. There are other methods that people use for those huge, dense populations. And here's an example of what Phragmites looks like in late May. So on to identification, some macroscopic traits. This photo over on the right, it shows what Phragmites looks like a little bit when it's a little bit more mature. The two subspecies of Phragmites australis look similar and grow in similar habitats. So I studied the subspecies descriptions carefully to make sure I could tell them apart. As with any attempt to identify a plant, it's best to study multiple trusted identification sources and observe multiple plants to account for natural variability and studying all the possible identifying characteristics. So here's some traits you don't need to be a botanist to observe. The invasive subspecies is taller, tends to have blue-green leaves instead of yellow-green. And at the end of the season, when the leaves dry up and break off, they tend to leave a little part of the lower leaf attached to the stem, whereas in the native subspecies, they tend to break off nearer to the attachment point. The culm, which is the stem under the leaf sheet, is rarely, if ever, red in the invasive subspecies, but often red in the native plant. The invasive plant grows in dense patches, crowding out other plants, and the native subspecies is a little friendlier, doesn't grow so densely and allows other plants to grow around it. The stems of the invasive subspecies are pretty strong and they're able to stand up under snow, but the native plants are not as robust, so they tend to bend over under snow. And finally, the invasive subspecies has become so common that it's possibly replaced the native subspecies colonies. So a closer look for identification. Here is over to the right some mature plants, and you can see the flowers up at the top, which are reddish brown. And then here is a leaf that I have stripped off a stem, showing the blade at the top, which goes off the photo several inches, and then the sheath at the bottom, which again goes off the photo at the bottom several inches before it attaches to the stem. In between the sheath and the blade is a structure called the ligule, and I mention that because you'll see that in some examples of how to tell the subspecies apart. Although I have read also that it's difficult to measure the ligule width, so I haven't included that in the characteristics I listed. Some of the identifying characteristics are not always reliable because they are overlapping. For example, both subspecies can have tan culms, and younger populations of the invasive subspecies may not be as tall or as dense as mature populations, so they could be misidentified by size alone. So I'm looking forward to seeing some native Phragmites. I haven't seen those yet, but I've learned of a couple places where I can go visit this summer. Fortunately, I'm friends with Art Gilman, research botanist at the Pringle Herbarium and University of Vermont, and I emailed him to help me make sure I could understand the difference between the two subspecies. And he told me the most reliable way is to detect tiny longitudinal ridges around the culm by running your thumbnail around the circumference. And the culms of native Phragmites are smooth and shiny. So here's a culm. I've stripped the leaf off, and you can see these tiny longitudinal ridges because I've enlarged the photo. And they cause light to reflect differently, so it's not quite as shiny. So it turns out that it's helpful to understand the life cycle of Phragmites in order to determine when it's best to harvest. So the plants start from seed in early spring. They also can start from stem fragments or overwintered rhizomes and then spread via the rhizomes that produce the vertical stems with alternate leaves and terminal flowers. Carbohydrates produced by photosynthesis in the green tissue fuels growth and are stored in the rhizomes. So harvesting during the summer, during the growth phase, arrests production of carbohydrates, thus deterring plant growth. And then because nutrients are translocated to the rhizomes at the end of the growing season, harvesting just before this translocation phase is really beneficial because it will deter next year's growth. If no harvesting is possible, removing the flower clusters before seeds disperse is a good idea because that can prevent spread by seed. So here are the two methods that were recommended that I tried. The first is called the spading method, and it's very simple but labor intensive. You take a sharpened spade and drive it into the ground at a 45-degree angle at the base of a stem. And then that cuts the stem below ground, and then you can lift the stems and collect them and dispose of them. So some tips for this method: it can be used on dry land or shallow water, and you can do it several times during the growing season as new shoots come up from the rhizomes. If you can only do it once, I mentioned before, best time is the end of the growing season. You're not supposed to try to dig out the plants because that will loosen the soil, making it easier for rhizomes to spread. Then proper disposal of cut plants is important because if you pile them up, they are liable to start sprouting and rooting. So you have to compost them carefully, preferably in a municipal composting facility where it gets really hot. Some people dry the plants, burn them, bury them, or store them in plastic bags until they're rotten and then can be safely composted. And that's the method preferred in Vermont. Although in Vermont, as a last resort, invasive plants can be bagged and thrown in the landfill. And then if seeds are present, it's best to bag the seed head before cutting off the stem so you're not inadvertently spreading seeds around. This photo is a screenshot from a wonderful video that Lynn Short produced, and I have a link to that on with my links pages. She is the one that developed this baiting method. She was a professor. She's retired now, and she's a very good teacher and has a lot of energy. She's still working on invasive plant control. So this just shows schematically what happens when you cut. These black marks are where you spade. And so you're just removing the photosynthetic tissue from the plant, and eventually the rhizomes will rot. So the other method is the cut to drown method, and very simply, this is cutting off the stems as close to the sediment as possible. And this is for plants that are growing in water. These raspberry cane cutter tools were recommended because they have such a small hook, that hooked blade, and so you can selectively cut Phragmites and leave the any native plants nearby untouched. So some tips for this method: cutting as close to the sediment as possible to avoid side shoot development. And so the deeper the water, the better chance of starving the rhizomes of oxygen, which is how this method works. And then I mentioned the cane cutter. You just hold the plant with one hand and with the other hand use your cutter to lop it off as close to the sediment as possible. And then being sure to remove all the cut material from the water because these plants will resprout from pieces. So here's the tools that I used. I originally used this smaller sickle last year because I was worried about the weight of the long handle of the raspberry cane cutter, but I did find that it didn't reach down to the sediment in a lot of places where the Phragmites was growing at Nichols Pond. So this year I purchased 2 of the cane cutters, hoping to find a buddy to help me do some harvesting. And I also used a spade. I borrowed one last year, but I bought a new one this year. And I did find that the tools needed sharpening after a couple hours. So, I brought some files with me. And other gear, gloves I found are essential for breaking up bunches of stems because there are some sharp places near the nodes. And then water shoes for spading in the mud I used, and a boat for the cut to drown harvesting. Bags for collecting plants, and of course, because we're using sharp tools, a first aid kit's important. Hat, sunscreen, water, and snacks. So here is a map of Nichols Pond showing places where I've observed Phragmites growing. I've numbered these chronologically based on when I found them. So number 1 is the first area I'd seen, and then number 8 I haven't seen, but Michael Gray's camp is near there and he's been harvesting, so he reported that. The Cahagan property is over here on the northeast side of the pond, and I chose sites 4, 5, 3 and 7 to do my pilot study. So I'm going to show you the results of my pilot study from 2 sites, 5 and 3. So here's site 5. I defined that as going from the outspan dock, which you can't see, but it's over here, to this beaver lodge. And I found I was overly ambitious to think that I could harvest this whole site. It turns out I could only harvest this small area from the outspan dock to a log over here. I think it's about 40 feet wide, maybe a little smaller, but at least in August, I was able to go in and harvest all the flowers from this entire site. So here is the biomass reduction that caused by harvesting. So I was able to harvest this site at the end of June, July, and August. And here's what the site looked like before I harvested in June. So as far as I know, there was never any harvesting going on. I'm standing on the outspan dock down here to take the photo. And then before the July harvest, when I came back, I saw that there was quite a lot of regrowth, and I was a little disappointed. But what I realized was that the lake level went way down last summer, and that caused the stems that I had cut. I had sickled these plants because they were underwater, and then in late July they were emergent, and that allowed oxygen to get in through the cut stems, and the plants, just like any cut grass, grew side shoots. So I went back and spaded all of these plants at the end of July, and in the end of August, I was happy to see that there was very sparse regrowth. Here's what the site looked like at the end of August after I'd done all my my spading, and I did do some sickling out in the deeper water. And there's the log that I used as my boundary. I found that there were some native plants in this area. I tried not to trample them. And I also measured an 8 by 8 foot area over in this location somewhere. I marked it off with flagging on this shrub. And I intend to continue counting and identifying the native plants and the Phragmites that grow in that area as time goes on. So successful manual control of Phragmites can happen, but it requires a commitment to keep harvesting. Apparently, after 2 or 3 years, there'll be very little growth. The rhizomes underground will have been starved. Just to show you about flower removal, which I did in that one site 5 at the end of August. Here's what the flowers look like up close, reddish brown, and very easy to cut off the tops of the stems. They're very, the circumference is small up there and the diameter. And so I could do that very quickly. So now moving on to site 3. This site had 2 little populations, and there's a cedar tree in between them. So over on the left is a more dense population, and that is where I tested my sickle out in May and showed you that photo of the early plants in May. And over on the right, there is a less dense population that I didn't even see in May. So those actually grew up a little bit later. I'm thinking it might be a younger population. So here's what starting with the site to the left of the cedar, here's what it looked like at the end of July. I harvested this site using the spading method at the end of July and the end of August. And then coming back at the end of August, here's what it looked like. So there was some growth from rhizomes, but it was sparse. And the only place I didn't sickle and do any harvesting was near this wasp nest over here. Hopefully I can get any plants that are underneath the wasp nest this year. Another way to look at biomass reduction caused by harvesting is, again, here's that photo from July before harvesting. And here is the same photo before harvesting in August. So there's not a lot of lake view lost. And here's the actual biomass that I harvested, big pile in August and a smaller pile in August, or big pile in July rather, smaller pile in August. And it took 3 3/4 hours less time to do all the harvesting. Now to look at that less dense site to the right of the cedar before the July harvest, I spaded all of these. And when I came back, in August, there were no plants there. So that one spading session deterred regrowth from the rhizomes that are underground there. That's why I think it might be a younger population. Okay, another way to look at biomass reduction. Here's all the harvest material from all the sites that I harvested in July and in August. So in July, there were 10 bags, in August, 2.5. And some sites 4 and 7 were quite small. There was no regrowth at site 4 in August. And it took 4 3/4 hours less time to harvest all the sites between August and July. So just to document the rest of the Phragmites along the Nichols Pond shoreline, here's a picture of site 2 in the end of July. So that site I have called going from this beaver lodge all the way over to the Erskine dock. And the Erskine dock is where I took those original photographs. And so this area right here that's in those original photographs from October of 2012 and 2015 is the most dense. It's been growing the longest, excuse me, in this area. And then it seems to have spread from right to left. I went down, every year I go down below the dam at Nichols Pond and walk along a trail up into the woods to visit some rare ferns. And I've been noticing that Phragmites has been growing along the edge of a wetland that's part of the Nichols Brook. And so that's kind of shocking, but there it is. And Nichols Pond is part of a 3-pond network of dammed ponds, starting with East Long Pond, then Nichols, and then Nichols Brook feeds into Macville Pond, and that feeds into the Lamoille River via Cooper Brook in Hardwick. So I haven't surveyed East Long Pond and Macville for Phragmites. I hope to do that this summer. Some observations. As I mentioned, down here in the lower left is a stem that I had cut when it was underwater, thinking I was using the cut to drown method. But then when the lake level went down, this stem was in the air and it put forth several side shoots. Whereas here, there are a couple of stems that I had sickled in deeper water and they remained underwater. And so you can see that this is what I sickled in July and down here in August. And there was regrowth from one node that was left above the sediment. So I'm hoping that with my longer handled cane cutter, I'll be able to get closer to the sediment. And over here, I just wanted to show that sometimes you get stolons growing above the sediment in water, and where light can get to them, chlorophyll develops. And so that allows for photosynthesis and carbohydrate production. They root at the nodes, just, I guess, looking for some substrate to anchor onto. And then here's just an example of some broken rhizomes that I happened to unearth. And because they're below the substrate, they lack chlorophyll because they haven't been exposed to sun. There was one native plant I was interested in. I at first thought that it might be a seedling Phragmites. So I looked at it more carefully and I found that as I drew my hand from the base upward, it was quite like sandpaper. And a couple of other, and Phragmites was smooth. And a couple other differences between this and Phragmites is that it had these downward pointing hairs at the nodes, and the stems were often zigzagged. So I'm not an expert in grass identification, but I did some homework and found that I thought this was cut grass, Liersia oryzoides. So I checked that with a botanist friend of mine, Jerry Jenkins, and he supported that idea. So just to end with some pretty pictures, these are some common plants around Nichols Pond that could be lost if the Phragmites populations were allowed to continue to grow. Coltsfoot over here, which is one of the earliest flowering plants in the spring and has my favorite Latin name, Tussilago farfara. And here's some aquatic smartweed, which grows in the shallows. And a lovely view of swamp candles. These lean out over the water and are easily seen when canoeing by. And then I was fortunate to find some horsetail strobili when I was up there in May. I love these beautiful geometric sporangia that produce the spores. I am keeping lists as I learn more about the plants growing there, especially the aquatic plants. So I'll keep ongoing lists of the plants I find. And then I just like to recap now some key points. Phragmites australis subspecies australis is very well adapted to becoming invasive, outcompeting other plants to create a monoculture, thus destroying biodiversity. A single stem's flower cluster can produce 1,000 seeds. Fortunately, they only have about 10% viability, and that does vary. But where you have a lot of stalks, that still adds up to a lot of seed potential. And the rhizomes can grow up to about 10 to 15 feet during one growing season, and pieces can generate a new plant. So some tips for harvesting. Just remember, it's the green material we want to remove photosynthetic ability, and thus the ability to store nutrients in the rhizomes. Now, sometimes spading was difficult because, especially on land, if there are roots or rocks around, so at least cutting off the stem at the base will discourage rhizome growth. It's important to even spade dead stems because the stem can deliver oxygen to the rhizomes. And the rhizomes develop next year's shoots in late summer or early fall. So spading at this time sometimes gives you a jump on next year's harvest by removing these shoots with a piece of the rhizome, maybe along with the vertical stem. Rhizomes grow more easily in disturbed soil, so avoid loosening the soil on land as much as possible. But Jan Gilbert from Ontario told me that we don't need to really worry about disturbing the underwater sediment. So when possible, you can pull up underwater rhizomes. The cut to drown method starves the rhizomes of oxygen. So it may result in shoot development below the cut. So especially in shallow water that transmits light. So it's best to cut as close to the sediment as possible and use the spading technique in shallow water. And I did find that Phragmites floats, so you can round up any harvested material that escapes. And then on to site recovery. Native plant seeds may be dormant in the ground, so removing Phragmites may give them the space to grow. So it's good to wait a couple of years before considering any plantings. So you give the native plants a chance to establish naturally. So I want to thank a bunch of people that helped me with this project. Jerry and Pat for encouraging me or inviting me to present this webinar last year, and Deborah for doing the marketing, and Taylor and Nick Meyer for allowing me to work on their land and disposing of many bags of harvested Phragmites. All of the control organizations for the work they have done and shared about online. and especially Vicky Simkovic and Jan Gilbert for answering emailed questions, and Lynn Short for developing the spading method and creating her information-packed videos, which I highly recommend. And then these incredibly talented botanists for plant identification help, Art Gilman, Jerry Jenkins, and Johanna Lagus. If you have any questions or comments for me, I'd love to be in touch via email. And my email is aofalton@yahoo.com. And then just to show you, these are the slides that you'll be able to access in the PDF file. The organizations, identification resources, manual control methods, and where to get tools. So thank you very much.
Wow, Annie, that's amazing, all your work, and also putting this presentation together. It's incredible. I'm going to just read some of the comments in the chat from Lynn. It would be a good idea to continue to document the amount of Phragmites harvested at each site over 3 years if possible, just to give you some more work to do.
Yes.
Annie, you're commended for all your work. You give me impetus to try this at Shadow, although we don't have these in the water, they are nearby. Yes, totally commendable work and documentation from Anne. I hope to join you this summer. Yes, hopefully we can get some people to join you because it is much more fun to do this kind of hard work with others. I hope other folks will be inspired to join you too. Much more fun to do with friends.
Yeah.
Couple more. Thank you for the detailed account of your work. Looks great. I have to leave, but look forward to seeing the recording and offer 2 observations. One, effectively sharpen my spade on my grinder, and it makes an amazing difference to have a sharpened one. And 2, placing harvested material on black plastic, then folding it over and leaving it in the sun for the rest of the season cooks and rots the plants and renders them unviable. I do this with poison parsnip too. I'll pause in case anybody has any questions to ask Annie, and then we can go back to the chat. So just feel free to unmute yourself if you wanted to ask Annie a question or comment.
Hi, I'm Susanna. I have a couple questions. I'll take my little hand down. I put this in the chat, so you can ignore it when we go back. But Annie, I noticed that you went 3 times, June, July, and August, and I'm gonna be trying to get some of my neighbors to work on a stand in our neighborhood. So if I were just gonna do 2 times, would you say like June and September, or what would you recommend for the 2 times?
I would say late July and late August or early September.
Okay.
That you want to get the plants harvested before that translocation phase. And I'm not sure exactly when that starts, but I found that at least at Nichols Pond, the plants were flowering at late August. So it seemed like that was probably a good time. I highly recommend looking into some of the research already done by all these great organizations on you know, in my slide about contacting those organizations. And the people that I contacted with, Lynn Short and Vicky Simkovic and Jan Gilbert, they were very friendly and helpful via email. So I'd recommend checking in with them because they're the experts. I'm just a beginner.
And their emails are in the slideshow or their contact
Yes,
info? Okay.
yes.
My other question, and I can definitely look into the other resources too, is did you come upon any information or yourself um, have experience like explaining to property owners why this was something that they would benefit from, like on their property, and kind of go through like how detrimental Phragmites can be?
That's why I'm doing this presentation.
Hmm.
You know, it's really geared toward people who own shoreline property. And, anybody like me who cares about biodiversity, and it enjoys, using lakes. Yeah.
Okay, thank you.
Yep. Spread the word.
I have a question too. This is Mary Waltz. I don't know how to put my hand up. I'm sorry, so I hope I can just interrupt.
Sure.
Yeah, thank you. Um, we have a project in Hyde Park which is focused on Japanese knotweed. So I'm listening to all this as a comparison to knotweed, and we want to start doing some Phragmites work. And I'm just curious why, and this is in relation to Phragmites on dry land as opposed to in the shoreline. Is it important to cut below the soil for Phragmites if you're on dry land as opposed to just cutting, sort of near to the ground above, like with clippers or sickle or something? Is there a reason it must be below ground on dry land that you're aware of?
Yes, definitely. And Lynn Short is here. Hi, Lynn. Thank you so much for joining us. And she's the one who developed the spading method. And so if would you mind answering that question, Lynn?
Yeah, first of all, Annie, you've done an amazing, comprehensive presentation.
Mhm.
You've put together so much information that's so valuable in one place. So I really am thankful that you are doing this, particularly,
Mhm.
talking to um, people in the United States where chemical solutions are quite often the first choice. And I feel like in natural ecosystems, it's so important to try everything except that, initially at least. So, Mary, did you say you're at High Park
Hyde Park, yes.
in Toronto?
No, Hyde Park in Vermont.
Oh my goodness, because I do a lot of stewardship in High Park in Toronto, and we're fighting Phragmites there at the pond.
Oh, right. Okay.
So
There are many Hyde Parks, it
I
turns out.
guess there
Yes.
are. I just thought, oh my goodness. Because I thought I haven't met you at the park. Anyway, it is my belief that spading below the ground gives you a better like if you're going to put the effort in by spading below the ground, you are uh, removing every possibility for photosynthesis. If you cut above the ground, that little stubble that you leave, still has the potential for photosynthesis. So it doesn't insult I always say we're insulting the plant when we do that. It doesn't insult the plant as much, like because you're leaving a little bit. And the other thing to remember, and Annie actually mentioned this, it's a grass. And when we cut the grass, we do that so that it will become more dense. So if you cut above the surface, you're effectively doing what you would do with a lawnmower on your lawn. And you're encouraging the plant to really get more dense. If you cut below the surface, you've stopped it from being able to photosynthesis, and then it has to mobilize the stored resources from deep in order to begin to grow again. So I think it is more effective. I mean, if all you can do is cut it above, at least you've taken up a lot of the resources. But I think if you want to sort of make the most of your effort, I would recommend going below.
Okay, understood. That makes sense to me, and I can understand that in relation to the knotweed experience we have.
And interestingly enough, Mary, I am currently Phragmites has been my focus for the last 20 years, but we have dog strangling vine, which is another problem plant, which I've done some research on manual techniques, and I have a publication on that one. And currently, I'm in the third year of working on manual strategies for Japanese knotweed. I haven't got anything published yet,
Okay.
but because it's a rhizome plant, the rhizomes are a different structure than
Phragmites, I'm taking this similar approach. I'm cutting above, I'm cutting below, and I'm pulling it out and I'm tarping it. But I'm finding everything you do to insult it is definitely curbing the growth. So
I'd agree with that. We've had good experience with manual control so far. We're in our 6th year this season, will be our knotweed program. But the other question I have, which is also comparing it to knotweed, is I understand that the seeds are, people generally talk about seeds on knotweed as not being viable or ambiguously not viable. You know, there's the seeds aren't the scariest part of knotweed. So, but I understand that Phragmites seeds are more viable, but they're not viable on the scale of, like, you know, invasive perennials. Oops, sorry about that. I understand you should get rid of the flowers because they will produce seeds, and you want to avoid that. But how scary are the seeds? I mean, are they you know, you want to just keep them contained in the site or contained in a container, but are they like on your clothing and you have to like defumigate yourself before you leave a site if you're in a site of mature Phragmites, or is there any information on that?
do you, Annie, would you like me to, I mean, you can chime in if you want, but the seeds for Phragmites when they're mature are very much like dandelion seeds. They're
Oh my God.
fluffy and they blow, which is one of the reasons why they are so efficient at spreading along highway corridors. You've probably noticed that in the ditches. So they, just general traffic creates wind and therefore spreads them along highway corridors that in Ontario where I am, that it's a real problem along the highway corridors. And then from there, they can blow into natural areas. So I think that that's why removing, when Annie was showing that image, those pictures of the flower heads, when they're the purple and they're not the big fluffy seeds, that's the best time to remove them because they are not blowing at that point.
Right.
Uh,
Right.
so they're not likely going to get on your clothes at that point. It should be just flowers and pollen. It's when they get to be those big fluffy seed heads
Right.
that they are, but they just blow in the wind.
We
So
do have some other comments in the chat we want to get to before we run out of time.
sorry, yeah.
Thank you very much. One quick comment I just wanted to say, because I think the bagging thing is a real problem for, this very effortful thing. And if we've used drying stacks for all of our stuff in Hyde Park for knotweed, and you could check out our website if you're interested in knowing more about drying stacks and how we set them up. Thank you very much. This is an excellent presentation and perfectly timed for us. Thank you.
Deborah?
Yeah, a couple more questions here. What did you do with the bags of plants?
I gave them to Nick and Taylor to dispose of, and they told me they did put them in the landfill. And, that isn't the best way to get rid of them, but for this pilot project, we felt that was appropriate. And I'm looking into some better ways to get rid of the plants this coming summer.
Okay, another question and comment. Thank you very much for your work and terrific presentation. Are you aware if any studies have been conducted using benthic barrier type treatment in locations of heavy infestation?
I am not. No, I haven't heard of anything like that in my work, my research online.
I think there's also another question, I think further up, to clarify again, the native subspecies has no ridges on the stem. I think if you can explain that again about the stem and the ridges.
Right. It's the invasive subspecies that has the little ridges on the stem, and you can't really see them. You really have to draw your thumbnail around the circumference to feel them.
Okay.
Alyssa has a question. Go ahead.
Alyssa? Unmute yourself.
Hi.
There
My
you go.
question is, has there been, this is all great short term, but you still have this very fertile field that is so easily repopulated by invasives such as Phragmites. Is there any thought to restoring shoreline vegetation with trees that, while they may take a long time to grow, will eventually bring about shade, which will help to prevent as many Phragmites as coming back since they need so much sunshine?
Yeah, I mean, what I have read about site recovery is it's perhaps best to let the seed bank alone and see what comes back. The work that Lynn has done has shown that the native plants come back quite thickly in some instances, and make it pretty difficult for Phragmites to get back in there. But also, these manual methods require some diligence. You need to go back every year and just check and see, how the sites are doing and if there's any more harvesting to be done.
There's a comment someone put in here that farmers might happily contribute a woven fabric bag for containment of material. This is what they get their sawdust in for the cows, better than plastic. So that's a suggestion of interest.
Thank you.
Deborah, do you see any others in the chat?
Well, I think we've covered most of it. A lot of grateful people for your presentation. And I just wanted to mention before people leave, because it's almost 1, that we will share a recording of this and Annie's presentation, which she created as a standalone document with live links to resources. We'll be sharing that on our website, and we'll do notifications on social media. And if you sign up for our mailing list, you'll get notified as when those are on our website.
Okay, so I will add my thanks, Annie. This was fabulous. I learned so much, and anxious to go back and read through all, go to all the links and read all the information, because this was really, really great. So really appreciate all the time you took to put this together. I want to thank Deborah and Jerry and the events committee led by our intrepid events chair, Jackie Sprague, who's here with us, and members of our board who put these things together. So thank you everyone for this. And as Deborah said, the recording will be up in a few days. And stay tuned. We'll be announcing our March webinar will be coming up. And also mark your calendars for Friday, June 5th, our lake seminar, which will be a hybrid all-day meeting. And hopefully a lot of people come in person. And we'll be announcing the theme of that in, I hope, a few weeks. So again, thank you, everybody, and we are adjourned.
Thank you.